Hello, I'm Beren Semiz, and I'm an assistant professor of electrical and electronics engineering at Koç University, Istanbul, Turkey. Today, I will present you our work on automatic subject identification using scale-based ballistocardiogram signals. Recent technological advancements have made it possible to embed various sensors into electronic devices, such as electronic scales, fitness equipment, and hospital equipment, which enable unobtrusive and non-invasive collection of physiological signals. These devices are often shared by multiple users, such as members of a family, athletes in a sports team, or patients in a hospital. Therefore, it becomes desirable that these shared devices can automatically identify their users based on collected signals. It is specifically important for improved user convenience, personalization of devices and services, as well as enabling safer and more secure systems. Currently, subject identification in most smart household devices, such as smart scales, rely either on simple biometrics, such as weight and heart rate, or require the user to manually introduce themselves by entering their ID or pairing with a third-party device, such as smartphone, at each time of use. The main drawback of state-of-the-art is that biometrics, such as weight or heart rate, are not subject-specific. In other words, they can change over time, and of course, different users may have similar weights. The drawback of the latter is its inconvenience. For example, the user has to carry their smartphone or enter their ID to the smart scale every time. In contrast, automatic subject identification using physiological signals removes both drawbacks, as physiological signals are naturally present in living individuals at all times, and they often contain subject-specific features. In this work, we studied subject identification using ballistocardiogram signals. At every heartbeat, the blood traveling along the vascular tree causes a small change in the center of mass of your body. In response to this change, body micromovements are produced by the recoil forces to maintain the overall momentum. Basically, ballistocardiogram signal measures these recoil forces of the body in reaction to cardiac ejection of blood into the vascular tree. Of course, ballistocardiogram signals are highly subject-specific. For example, here we have signals from 3 different subjects, and as you can see, the number of peaks, durations between consecutive peaks, amplitude values, etc., they are all varying among these 3 subjects. So overall, we can safely say that ballistocardiogram signals can provide an unobtrusive and variable-free method to distinguish between different subjects. Basically, ballistocardiogram signals can be acquired using beds, chairs, wearables, and modified scales. In particular, in this study, we used a modified weighing scale to measure the BCG signals. And simultaneously, electrocardiogram signal was being recorded. The subjects underwent the following procedure. First, there was a 5 minutes long initial rest period. And this rest period was followed by 20 seconds of Valsalva maneuver. And after this Valsalva maneuver, subjects were again at rest position for 2 minutes. This rest period was followed by walking exercise for 2 minutes and squatting exercise for 90 seconds. And again, we had this 5 minutes of rest period. Finally, the subject's left hand was immersed into cold water for 15 seconds. And in the end, again, we had this 2 minutes of rest period. We only used the rest periods in our analysis when the subject was on the scale. Thus, both the ballistocardiogram signal and the electrocardiogram signal were available for us. Also, the model was trained using only the initial rest recordings, so the rest periods which follow the physiological modulations were only used in the final experiments where we tested the effect of physiological modulation on subject identification accuracy. So let me briefly talk about our preprocessing and beat detection pipeline. First, of course, the signals were filtered, and following that, we proposed two options for beat detection, one with ECG assistance and another one without ECG assistance. For the ECG assisted version, R peaks were detected on the ECG signal, and the BCG signal was segmented into beats using these R peak locations. The beat length was determined to be 600 milliseconds as previously done in the literature. These BCG beats were then ensemble averaged to remove noise and reduce the impact of outlier beats. The moving window size was determined to be 3 beats per frame with an overlap of 2 beats between consecutive ensemble averaged frames. On the other hand, for the non-assisted version, we used the J peaks on the ballistocardiogram signal as our reference. To detect the J peaks, the ballistocardiogram portion where amplitude was greater than 0 was taken on the BCG signal, and the upper envelope was constructed. Basically, the local maxima points in this envelope were corresponding to the J peaks. Once the J peaks were located, we took the BCG signal segments that are 250 milliseconds before and 350 milliseconds after each J peak location on the BCG recording. So in total, our beat length was again 600 milliseconds. The detected beats were then ensemble averaged into frames using a moving average window size of 3 beats per frame, identical to the first case above. Following this, from each ensemble average frame, we extracted some features. And for this feature extraction step, we mainly focused on the IJK waves, which correspond to the blood ejection waves. So in total, we extracted 12 features, and these features were amplitudes and locations of IJK waves, durations of IJ, JK, and IJK segments, RMS power of the IJK complex, and amplitudes of IJ, and JK waves. And let me briefly talk about the classifier training and prediction part. Basically, we pose the subject identification problem as a multi-class classification task that can be solved via supervised machine learning. Basically, each instance in the training data was corresponding to an ensemble average BCG frame and was of the form XI, YI, where XI were the 12 features extracted and YI was the label equal to the unique subject identifier. We chose support vector machine or SVM model since it's a popular supervised learning method in biometrics and bioinformatics due to its accuracy, flexibility, and ability to deal with high-dimensional feature spaces, as well as high performance in physiological signal analysis. We implemented support vector machine with grid search to automatically search for optimal hyperparameters. And our model was supporting multi-class classification through the one versus all strategy. At prediction time, an unlabeled BCG recording was provided to the system, and a set of unlabeled feature vectors were obtained. Using these feature vectors, our model predicted the label for each vector, or let's say for each ensemble average beat within the test recording. And the predicted subject ID label for this test recording was obtained by calculating the label that was observed most number of times. In our system, each prediction was, of course, associated with a confidence value, denoting how confident our system is in predicting the true subject ID of a test BCG recording. And in order to calculate the confidence value, we calculated the ratio of the number of occurrences of a predicted label among the beats and total number of beats for that specific test recording. We use a threshold tau such that for a test recording with confidence less than this tau value, the system outputs that subject identification was unsuccessful for that recording. In other words, subject could not be found. However, if confidence was greater or equal than this tau value, then the system was outputting the Y label prediction as usual. So let me briefly present you our our results. And the first one is the results we obtained for subject identification accuracy. In the experiments, we operated in multiples of 20 seconds since it's a reasonable amount of time to stand on a smart scale for body mass index, vital signs, and fat and water percentage measurements. We divided the continuous ballistocardiogram and electrocardiogram signals into 20 seconds long segments to obtain multiple non-overlapping recordings per subject. We used leave K recordings out cross-validation. In other words, we ran multiple iterations where in each iteration we left out K recordings of each subject from the training data. We then tested the model on these held out recordings. The number of K was, of course, changing our test and training durations. We performed several experiments. In the first one, the test recording duration was fixed to 20 seconds, and we experimented with total training duration varying between 20 seconds and 280 seconds. And in the second one, the total training data duration was fixed to 1 minute per subject, and we experimented with test recording durations varying between 20 seconds and 240 seconds. Here are the key results we observed. The first one is increasing the training or testing duration increases the subject identification accuracy. And the second one was that ECG assistance increases subject identification accuracy compared to the non-assisted version. Following that, we performed some tests for the prediction confidence value. So we varied the confidence threshold between 0 and 1 in increments of 0.1 and measured the mispredictions prevented as well as the correct predictions lost under different values. Here we have results for two different training durations, 20 seconds and 1 minute with and without ECG assistance. In each setting, two lines were plotted, the ratio of mispredictions prevented and the ratio of correct predictions lost. We observed that tau less than 0.3 causes little or no loss in correct predictions, while being able to prevent some mispredictions. On the other hand, the value of which maximizes the difference between prevented mispredictions and loss of correct predictions was around tau equals 0.6. And basically, this was the result we used in most of our reported results. In addition, longer training durations not only increased the confidence in correct predictions, but also decreased confidence in mispredictions. And as our final experiment, we investigated the effect of physiological modulation on subject identification accuracy. As you may guess, in some cases, it's also possible that subjects are involved in some form of physiological exercise before they step on the scale, which increases their heart rate and results in non-traditional ballistocardiogram and electrocardiogram signals. Thus, we also measured subject identification under physiological modulation. Basically, we used the rest recordings taken after each physiological modulation. And we again performed our experiments both on the ECG-assisted and non-assisted versions. Overall, we observed that the accuracy of modulated recordings is 15 to 20% lower than rest recordings. This drop was actually expected, considering that the training data did not contain any modulation. And also, we observed that the type of modulation was also important in model accuracy. So thank you, and if you have any questions, I'll be happy to answer.